stealth technology, the internet, GPS in the palm of your hand, autonomous operation.
Technology is a driver of our times. Since its founding in 1958 in the midst of the Cold War, DARPA, the Defense Advanced Research Projects Agency, has been a driver of technology. Welcome to Voices from DARPA, a window onto DARPA's core of program managers. Their job, to redefine what is possible. My name is Ivan Amato, and I'm your DARPA host. And today I'm pleased to have with me in the studio Renee Wegrzyn, a program manager since early 2016 in DARPA's Biological Technologies Office. Renee earned her bachelor and doctorate degrees in the field of applied biology from the Georgia Institute of Technology, and then did postdoctoral work in molecular biology in Heidelberg, Germany, as an Alexander von Humboldt Fellow. A strategic research driver for her is to apply tools of synthetic biology, which draws from genetic engineering, molecular biology, and other arenas, to support biosecurity and to outpace infectious disease. Prior to joining DARPA as a program manager, Renee was a senior lead biotechnologist at Booz Allen Hamilton, where she led a team that provided scientific and strategic support in the areas of biodefense, biosecurity, disruptive technologies, emerging infectious disease, neuromodulation, and synthetic biology to DARPA and other federal and private institutions. She also led research and development teams in the biotech industry that focused on the development of immunoassays and other tools for molecular biology and disease diagnostics. Renee, it's my pleasure to have you in the studio today.
Thank you, Ivan. Pleasure to be here.
Let's start with how you got interested in science and technology. What was it? Was there a teacher that transformed you, that turned you on to science? Did you come from a scientific household? What was it?
It's hard for me to think of a time when I didn't have science in mind, even from a very young age. So growing up in Florida, we always had access to little critters on the beach or in little creeks in our backyard that we would play with. And my sister, her name's Lisa, she's 11 years older than me, and she had a career that was really on a trajectory to be a scientist until later in life she switched to be a lawyer. But in the early days, I got to go to the lab with her and kind of see what that world looked like, and that got me really interested. And so from a very early age, in high school, I was a lab assistant to one of the teachers where I helped him embalm some of the animals that we would dissect in classes. I think I was very lucky through elementary school and high school, and then going into college and beyond. I always had opportunities to be really hands-on with the sciences, to understand what types of science I really liked and what I didn't like. And I remember in high school, one of our teachers would take us to a local, I think it was a limestone mine in Florida, where they would pump out dirt and rocks from really deep into the earth. And there was many fossils, mastodon teeth and things like this that you could find. And that really had an impression on me, because that's where I first started to learn about evolution. And then all through school, I did take the opportunities to work in the laboratory as an undergraduate at Georgia Tech, and that's why I came back to do a PhD, because I already had about a year and a half under my belt.
Right, and for listeners who have never seen a mastodon tooth, I actually have. I mean, they're like the size of erasers. They're just amazing things to see. Obviously, that was catalytic for
right.
you. And when I look at your degrees, both of them are in applied biology, which is to say your bachelor's degree and your PhD. I don't know if there's a master's along the way, but both in applied biology, which tells me that from a very early age you were looking at biology as technology, perhaps. So how did that early insight come to you?
That's an interesting way to think about it. I hadn't thought about it that way, but I did always see biology as a tool, and a tool to understanding the world around us. And later then in university, it's a tool to understand disease, and then be integrated actually even with a person moving into the medical device world. At Georgia Tech, I was in one of the early classes that was working with the bioengineers. They were just launching a bioengineering program at Georgia Tech. So it was really interesting for me to interact with the engineers who would actually build a device based on things like cellular mechanics and fluidics. And that was pretty mind-boggling to me that you could take that information and actually turn it into a device to improve people's lives.
So for some years, you worked as a contractor with Booz Allen Hamilton, and that's a company that's a lot of work with government agencies, including DARPA. Is that actually how you got to know DARPA?
No, actually, I got to know DARPA when I was working in the biotech industry up here in Rockville, Maryland. And the CEO of a small company I worked at called AdLife, this is where we were developing diagnostics for neurodegenerative diseases based on protein misfolding and indicators of disease. And the CEO of that company, Alan Rudolph, used to be a DARPA program manager. And I had not heard about DARPA before meeting Alan. So sometimes over lunch or just casually, he would talk about this magical place called DARPA, where it really seemed that you can have a wild idea, and as long as you had a great technical plan to make it happen, there could be a path forward to try to implement that. And the more I learned about DARPA, the more I really got interested. And when I was looking for the next step in my career, Alan, knowing me well, I just asked, what do you think could be something that would be good for me? I like the pace of the startup environment, but job security would be a little bit better. Longer-term job security would be of interest. And so he suggested that I look into different types of contracting and consulting agencies around the D.C. area to support agencies like DARPA. So there's multiple ARPA-type agencies. And luckily, I just found a great position with Booz Allen and stayed with them for quite a few years as a CETA, not only at DARPA, but other places like IARPA, the Intelligence Advanced Research Projects Activity, and the Bill and Melinda Gates Foundation as well. I helped them with some of their early grand challenges.
Right, and you used a little government speak in there,
yes.
which is CETA. But I want to stay on that for a second. That stands for Scientific Engineering Technical Advisor.
That's right.
That's actually a word we hear a lot around here. There are more CETAs here than there are program managers. You are now a program manager. Mm-hmm. So you've actually worn both hats. And I'm wondering if you can just talk a little bit about those two categories of employees here, the CETA and the program manager.
Mm-hmm. As you just mentioned, there are two categories of employees here. There's the government employee, which is the program manager and other supporting staff, and the CETAs. And the way I like to describe that model to folks in biology especially, they're familiar with having a PI in a lab and then people that work with them to support their vision, right? A principal investigator in a laboratory. They have a supporting staff of, they could be grad students, they could be staff scientists to help them implement the vision of that PI. And really, DARPA, to me, the program manager and CETA role is very much along those lines. The program manager is responsible for the vision of the program and the direction forward and the strategy. And the CETA team serves various roles. They can provide technical content that's complementary to the program manager. They may have strategic guidance. They may have deep DARPA experience, because oftentimes a CETA may be here for many years longer than a program manager, who, of course, we're here for these short-term appointments.
Let's now sort of get into your DARPA life a little bit. Mm-hmm. In 2016, you became a program manager. At that time, you inherited one program called Living Foundries, which is about engineering the exquisite control that living cells have over, say, atoms and molecules, but in this case for technological ends such as new materials, industrial chemicals, pharmaceuticals, and agricultural products. And then you developed a program called Safe Genes, which you were able to get through the very difficult vetting and approval process here. And this is all about developing a toolkit to rein in, limit, and even reverse some of the emerging and extremely powerful tools in genetics and genomics and molecular biology. I want you to talk about those, but before we do that, I just want to hear a little bit about how you think about biology, because as we look at some of these tools, and we'll talk about them a little bit, like CRISPR-Cas9, to me, they're sort of miraculous little nanoscale machines, and it makes me, sort of look at life with a kind of reverence and awe. I don't mean to lead you; that's not nice of me. But talk to me about how you view biology on these sort of molecular scales and even higher scales.
The way I think about biology is really from an applied approach, and that has a lot to do with, I think, having been brought up scientifically at Georgia Tech, always immersed in not only biology but surrounded with engineering. When I look at a biological system, I think about how that system could be manipulated in a programmable way. Looking at some of the tools that synthetic biology brings to us, or genome editing, like CRISPR-Cas9, which is CRISPR is one of the biggest acronyms we'll use today. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. What this protein does, it's really interesting. The origins are it was a bacterial immune system effectively, where a phage virus would insert its DNA into a bacteria. That bacteria would cut up that DNA using CRISPR-Cas systems and take little pieces of that phage DNA and put it into its own genome as a memory of that infection, so that if that virus came back again to infect that cell, it would say, oh, hey, I have this information that I've seen this before. I can now neutralize it. So back in 2012, what researchers Jennifer Doudna and others recognized were you can actually program what Cas9 recognizes. It doesn't have to be a phage, but you can provide it with the Cas9 with the information to say, okay, go after this specific piece of DNA. And that was this very powerful, what I like to think of from the DARPA perspective as a Sputnik moment in biology. So we had a lot of innovation in DNA sequencing and synthesis. You could look over the decades that we kept improving that technology. It's very powerful now. But all of a sudden, we had a tool to be able to edit that DNA. And that's really has changed a lot in the field now. There's very few organisms that can't be edited.
I love the Sputnik moment, partly because when you look at some phages, one is named T4, they look like little spacecraft.
Right.
So this is more apropos than maybe you knew.
That's right, yeah. There's even microsatellites in DNA sometimes. So. But for me, that's what created an urgency for this new tool in biology that I think makes that national security connection for me.
So let's talk about the two programs. We'll start with the one that you inherited, Living Foundries. Tell me a little bit about the history of that and its status.
Sure. So the original program manager who launched that effort, Alicia Jackson, she had this vision to start to create a tool bed where we could distill biology to an engineering practice, where you could take something like the design, build, test, learn cycle of engineering and apply that to a biological system like a microbe, an E. coli or a yeast or other type of bacteria, to make products and molecules that would be of value to the DoD. And I really think that her inspiration came from looking at biology as this incredible system that could make a real diversity of molecules that extends well beyond what we can make through traditional manufacturing and chemical processes. So when you have access to that diversity of biology, can you start to manipulate that to make new molecules, things that have never been made before by biology, which are some of the most ambitious goals of the program. So her foundational investments really created the tools under each one of those categories, the design tools that you can quickly in an automated way design a biosynthetic pathway to stitch genes together to put into a microbe. Build, so this is the DNA synthesis and insertion of DNA into a living system. Testing, this is the mass spectrometry and other measurement tools to look if a product's been made. And importantly, learning. So this is some of the designs you make are not going to work, and programming that into your learning cycle to not use that design and make a better design next time is really important. So when I took over the program, the Living Foundries 1,000 Molecules program was just taking off. The first phase of the program was to build those individual components and tools, and now the second phase is to really integrate them as a platform to create this foundry-type capability that the U.S. military or a customer, a business customer, could go to a foundry and say, I would like you to make this molecule. Can you develop the biosynthetic pathway and the organism to do it? So the ultimate demonstration of that capability, that foundry capability, is to make 1,000 unique molecules. And we're 551 molecules in. So our teams, which are Amherst, Zymergen, and MIT Broad Institute, they've collectively made 551 molecules.
And can you just give listeners a sense of the kinds of molecules
we're
talking
Sure. The kinds of molecules, so they're very diverse. They can be anything from adhesives to coatings to paint you might want to put on the outside of an aircraft, all the way to small molecule pharmaceuticals that could be used for medicines, countermeasures, cancer treatments, you name it. The whole gamut of small molecules are being developed under that program. And now we're about 2 years into a 4-year program. The final 2 years is going to prioritize a small subset of those molecules that have been produced that are very compelling that we'd like to scale up. So far we've made pretty much detectable amounts, meaning very small amounts of each one of those molecules, and we'd like to move to the kilogram level so that these are now testable amounts that we can really implement in larger experiments and testing.
Yeah. Absolutely exciting area. I mean, when we think about biology, I mean, I think of things like a sequoia forest, an Amazon forest. I mean, the diversity of creatures and then all of the tissues that they're made of, all of the materials they're made of, hormones and neurotransmitters, all of these powerful kinds of substances come out of biology, and now here bringing a different mindset, but with that same kind of context of synthesis. Really kind of an exciting area.
I think it's a great point that you mentioned that biological diversity across the globe, because what many of the teams are doing are looking at extreme environments across the globe, looking at microbes, looking at plants, even looking at some human enzymes, and you can stitch those together, a plant enzyme, a human enzyme, some extremophile microbe enzyme all together in a workhorse organism in the laboratory to make that. So this program is very much dependent on biological diversity and looking at the enzymes that can be detected in the wild, doing their normal jobs that they do in the wild, but we repurpose them in the laboratory.
Right. So you're also leveraging the way evolution has pushed biology to be innovative in its biological context, sort of seeing where you might find useful things there, but then building even further with human technology.
Absolutely.
Yeah. So then let's go to Safe Genes. This was your first baby here, right? So talk to us about that, the origin of it in your mind, and what the status of that program is now.
Sure. So Safe Genes is a program that's the program I came to DARPA to run. And for the listeners, if they don't know, the way that a program is stood up at DARPA is it's a very grassroots type of process where it comes from the program manager, an initial idea or concept that we can bring to our leadership and to the director of DARPA that gets developed then into a program. And so I came to DARPA with the idea to really try to harness the beneficent use of genome editing technologies like CRISPR-Cas9, but to prevent their potential for misuse. So at this point, these tools have really been democratized. It's arguable that probably every molecular biology lab in the world has access to these tools and is using these tools. So the genie's out of the bottle, it's out there. And there's a lot of power in these tools for editing genomes, and we can do a lot of really tremendous good with these tools, but their potential for misuse, both nefarious and accidental, is a real vulnerability that I came to the agency to try to address. Not only the ability to control editing activity, if you want to use it in a given system, to limit it only for use in that system, to counter. At the start of the Safe Genes program, there was no countermeasure available, meaning no inhibitor of a genome editor that could shut down if its activity, if it should accidentally be misused or in a more malicious misuse. And then finally, if a change is made, how can we restore a genome back to its baseline state to remediate any type of genetic change that may be unwanted?
Yeah, it's a remarkable set of tools. One of them that I heard you talk about reminds me of the telomere, which ticks off how many times a cell has replicated, and there's only so many times that a cell can do that before it dies off. It sounds to me like there's something like that you or your performers, and by that I mean the research groups that have contracts under the Safe Genes program, that they might be working on some similar mechanism to limit a genetically edited cell in its just how long it can be out there.
Mm-hmm. So that would definitely be under the category of a controller that we're developing. We do have some generation counters. So one thing that I haven't mentioned is there's derivative tools that are made from genome editors called gene drives. And this is one of the most powerful uses of these technologies, because what gene drives do is they ultimately bias the outcomes of inheritance, where when mom and dad get together, you normally have a 50-50 chance of a gene in an offspring coming from mom or dad. But through a gene drive, when you couple a gene or a trait with a genome editor, that can force its inheritance to near 100% efficiency. And so when you can bias the outcome to levels that reach up to 100%, you want to be able to have a way to control that if you don't want that spread of that trait to go on in perpetuity. And so one way you can do that is through a generation counter, that if you want to keep something like a gene drive local, you may say, okay, after 5 generations and reproductive cycles, this can shut down. And so these are some of the technologies we're starting to explore. Right. So clearly you are thick in an area of biology,
powerful biological technologies, and some of those who have been in from the beginning. I'm thinking here of Jennifer Doudna, who has been in on this from the beginning. So she's written a book that I read recently, and when you read that, she is both utopian in thinking about the power of tools like CRISPR-Cas9 in curing all kinds of diseases and doing all kinds of good things, but she also is at some points in the book sort of dystopian. You are in the middle of this. When you go out to conferences, the promise and the peril of these technologies are always part of that discussion. How are you thinking about that as someone who is in a position now to direct to some degree how this research goes?
Mm-hmm. So somebody has once described me as surprisingly upbeat for someone working on gene editing and national security. But I'm really excited about the promise of these tools, not only for addressing inherited genetic disorders, but the potential for use of these tools to eliminate infectious disease in a way that's superior to our current countermeasures that we use. And I look forward to the opportunity to be able to test those tools. I think there's a lot to be said for biodiversity as well, in terms of a tool like a gene drive is going to have to face natural biodiversity. A perfect gene drive would only work in a system where an organism's genome is identical all the way across the globe, and that just doesn't exist in any system. So I think in reality, some of the potential for misuse of these tools is actually going to be very challenging to achieve. And as long as we're addressing those risks while we develop the beneficent use technologies, I think we could be on a good, safe course forward.
When you think about the work that you are doing now in the Biological Technologies Office, in DARPA as an institution, what can you say about DARPA's place in the overall kind of innovation ecosystem? Because you've worked in a number of parts of that ecosystem, so I think you might have an interesting view on that.
Mm-hmm. Having been at DARPA now in some capacity for about 8 years, I can absolutely say DARPA is leading edge, and it's a place where you learn about new technologies, new ideas. We have people from industry, from academia reach out to us all the time with new concepts. So I do feel that DARPA is a special place where we have early access to these technologies, or at least the information about these technologies, and really a responsibility to look at them and understand, how could these tools be used for good, used for bad, and really be ready to address both of those in an appropriate way.
And when you think about tools like CRISPR-Cas9, you spoke about this tool within the context of the programs you're working on. But when you think of maybe the best application that you could imagine down the line, mm-hmm. what comes to mind?
The answer to that question probably varies depending on who you're speaking with. So I've had the opportunity to be in meetings with patient advocates for a number of inherited genetic disorders, and the ability to create a therapeutic for an adult individual who has suffered long-term from a disease, I think is incredibly exciting if we can do something to improve that individual's quality of life. They're desperate for those technologies, and I think here's an opportunity for a tool to do a lot of good. So that's one of the things I'm most excited about. And many diseases that otherwise have no treatment options at all. They're really just treating symptoms, trying to make patients more comfortable. And the other is, of course, this is one a lot of people point to for gene drives, but is the ability to eliminate vectors that carry diseases, things like malaria or dengue or chikungunya. As we have more and more emergent diseases that are spread through insect vectors, this would be an incredible tool to implement to prevent their spread to humans.
Right. And again, though, we're talking about such powerful technology. So in the debate here has been, do we get rid of the mosquitoes, the vectors that actually carry these things, which sounds on the surface to be, that sounds like a solution. But on the other hand, and I think you've talked about this yourself, we are talking about very complex ecological environments. And so even though we think about mosquitoes as essentially bad things, we don't know if we got rid of them, what would that do ecologically, right?
Right. Yeah, and that's something that we have to take a very serious, close look at, because we could be opening up a new ecological niche, right? If we eliminate one species of mosquito, And there are many species of mosquito out there, right? So you can be inviting a new mosquito to fill in that ecological niche and potentially bring in a different disease. And so really, Safe Genes is a short-term funding for 4 years. There's no open release, but we want to start to develop test beds for these systems to understand how they behave over generations. But I think this question of how this might work in the environment and in an ecosystem, won't be answered in the next decade or next decades until we can really understand how these systems.
Right. It's interesting. I often think about the history of technology as sort of the solution becomes the problem. I'm thinking here of things like CFC, chlorofluorocarbons, which were a great solution to a refrigeration problem early last century. People were dying from the refrigerants that were being used at that time. CFCs come in, they're not flammable, they're not toxic, and they become these wonder chemicals for that purpose. But then as they're made in massive amounts, the same stability that makes them great goes up to the stratosphere and starts to eat away at the ozone. So they become one of the great chemo villains of the 20th century. When you think about biological technologies in this context of the solution could become the problem, what are the problems that keep you up at night?
Mm-hmm. Well, if we go back to the gene drive examples, it is that, what are the long-term implications? And so this would be a genetically modified organism released into the environment. And if we have to be sure that we carefully consider how one might use these tools, it's not going to be a silver bullet for every problem. We shouldn't have 50 different types of mosquito species engineered with gene drives. It's going to turn into a space junk type situation where we don't know how these individual organisms are going to be interacting with each other. What is the long-term effect in the environment? This is, I agree. I don't think that's a long-term solution. And for me, part of the Safe Genes program and just the message that I'd like to get out there is to have that long-term thinking in place where even in the initial designs of the experiments, what might this look like in 10 years, 20 years, 30 years, well beyond the end of a short DARPA program. So we can build in some of those safety switches early on at the inception of a technology.
It's heartening to me to know that you as a program manager here are thinking about both those pros and cons, the promise and the peril of these powerful technologies. And it's going to be exciting to see where you take your existing programs and maybe some other ones that you'll get started in the near future. Renee, is there a question that you wish I had asked or you wish some of your other interviewers from other interviews in the past had asked you that I haven't? And if so, you can ask it and even answer it right now.
One of the questions people do ask me, and I want to put a plug here for the iGEM competition. So this is an International Genetically Engineered Machines competition. This is a place where a lot of new researchers and individuals, amateurs interested in synthetic biology get their start. This is a pretty low threshold way to get into a competition setting to say, what's something cool you can do with engineering biology and synthetic biology? And the organizers of this competition have always, first and foremost, put biosafety and biosecurity as part of these programs. Each of the teams, these young students, these undergraduates, sometimes even high school students, have to address safety and security in all of their designs. And wouldn't that be a great thing for every lab to be able to do? One of the things I've noticed since being a program manager is that people are often blind to the potential misuse of their own technologies. It takes somebody from the outside to be able to look in and see where those potential vulnerabilities are. Not only for just the system won't work, it might break, to, wow, this could actually become a problem. You mentioned biotechnology becoming the problem. I think if we had the ability to really look objectively at each other's work and try to identify those vulnerabilities and then mitigate the risks that are presented, I'd be much more excited about a path forward for biotechnology.
Okay, well, I think we're about out of time, so I just want to thank you for being here. This was an amazingly interesting discussion for me. Thanks for sharing these stories about how you came to science and what's motivating you here as a program manager. So thanks for being here.
Thanks, Ivan. It's been a pleasure.
And thanks, listeners, for sharing this time with us. I hope you join us again for the next Voices from DARPA. For more information about Renee Wegrzyn, the programs she runs, and the other breakthrough technologies DARPA is working on, visit darpa.mil. And for links that enable you to download this podcast, go to the Voices from DARPA page on DARPA's website.